So thank you very much for listening today. My name is Stephane Fadanka, and I'm so happy to be here with you to share our journey so far in the very difficult and challenging aspect of biotech entrepreneurship. I think today I'll be very happy to also have a lot of questions from the participants and also a lot of feedbacks as well after entertaining everyone on what's going on and what we do here in Cameroon. So my name is Stephane Fadanka. I have a training in molecular biology. I'm the executive director of MboaLab Biotech. I also work and also a member of some organizations such as Symbol Africa, Open Science, Open Life Science, and also RET program. So you can reach out to me through the various links, email. So today, really, I'll be very happy to share with you our journey so far in the biotech entrepreneurship, and also talking about the various realization and achievement we've been able to do. Also mentioning some of the key points to focus on in case someone is also interested in doing the same thing. And then at the end of the day, it will be very interesting to have a Q&A session. So biotech entrepreneurship, I would like to start by like kind of redefining some terminologies and also to provide some basic information about what it's all about, because basically what we are doing is biotech entrepreneurship. So literally, it refers to a practice of identifying, developing, and commercializing innovative solutions and products. So really, in biotech entrepreneurship, you really need to target and identify specific scientific innovation, like and also go through the process of developing and commercializing it to a concrete product. So usually, when you talk about biotech, it involves using living organisms, which can be cells and other biological systems, to create or improve products and processes that have application in various industries. So really, there's two terminologies here. There's the biotechnology side that deals with identifying, developing innovative solution in the biology, and also the entrepreneurship aspect of it. So what biotech entrepreneurs do, they leverage scientific advancement and business acumen to transform research and development into tangible products. So it's very important because at the end of the day, what you need to have is a concrete product that solve particular issues or address a particular problem in a concrete manner. So I want to draw the attention here that the product can be a physical thing, something really concrete, and it can also be a service. So there's a distinction to do here, but basically you need to leverage scientific advancement and also adding the business acumen to solve particular product with a concrete product. Now, I would like to talk about some key points to really keep in mind when you want to go towards this journey. You need to think about innovation and research. So if you want to start a biotech enterprise, you really need to think about the innovation. Usually, the innovative aspect come from research and development. So you really need to think about scientific or technological breakthrough or discovery. So it can be drugs, diagnostic tool, genetic engineering techniques, so something new. And then that would serve as basis for your business proposition. Now, after that, once you have a breakthrough or a scientific discovery, you need to think about the business side of it. So you have to create a comprehensive business plan, outline the technology potential, target to market. So now the plan will guide you through the path of concept to market. So it's important And usually, as scientists, we are very good at doing the point one, and now we don't have the training for the business planning. So it's very important in the team to have really complementary people who can look at both aspects or different aspects of starting the business. So now then you need to think about funding and investment. Usually to start up like any other business, like even if you want to have a shop or any other business, you need to think about the funding side of it. How do you get the resources to build the infrastructures, like pay the salary of the team? So usually it requires substantial funding for research development. In general, after having the idea, you also need to push again to refine the discovery so that you have a concrete product. So it's done through research and development, and also a process that takes some resources. So you need to think about that. Seek funding from various sources. I just highlighted some here, but there are many different sources, as we can see, as we will see in the next slides. And then another very important point is regulatory and compliance. So biotech entrepreneurship deals with navigating the complex regulatory landscape, because you need to make sure that what you are doing is kind of framed under first the country regulation and also international regulation. So we really need to be aware of that. Most often, it deals with very advanced scientific experiment. So you need to make sure that what you are doing is not harmful, or you need to know exactly have a strong notion on the regulatory landscape within your own context. So these are just examples. Now, once you have that, you also need to look at the market entry and commercialization. So you have to draw strategy for launching your products. They usually involve identify target audience, pricing strategy, distribution. So a good trick here is really, while you are developing your innovation, it's good to involve the end users right from the beginning. Usually it's a process that help boost and improve the adoption, because at the end of the day you have a product. What guarantee do you have that the product will be accepted by whatever end users you are targeting? So it's very interesting and important to also involve people right at the beginning. I'll mention some strategy on how to do that, so that you are sure that after passing through all these stressful steps, people do accept and use your products. So now we also have to talk about partnership and collaboration. So most often it's very important and it's a critical aspect of starting a biotech company. Most often you have to collaborate with research organization, universities, like private organization, to leverage on the expertise and the resources. Most often it's very important. So these are just in a nutshell summarized what are the various steps you need to keep in mind when you talk about biotech entrepreneurship. It's not an exhaustive list, but for me, is these are the main points you need to watch out. So now I would like to take our own experience as case study. So how how did we follow all these steps to come up with a concrete company, concrete products? And I would like to start by defining or presenting the organization. So I'm part of the MboaLab. It's a Cameroon-based innovation hub founded in 2017. So our real objective, our mission, is to catalyze local sustainable development through open science, and we do that through different activities. We focus on training and capacity building. We develop open educational resources and also some hardware prototypes, because as we are going to see, access to biotechnology is also tightly related to access and availability of equipment. So it's very important to be able to also see how to produce your own equipment. We are also involved in active research, international research. and also the main thing that we started was the open enzyme biomanufacturing. So I'd like to talk a bit more about the activities. At early stage, since 2019, soon after the creation of the organization, we've been able to do the local production of enzymes and reagents. So this was to solve a concrete problem that is still an issue in Cameroon, the access to reagents, especially in application. So we really wanted to tackle that issue and provide research institute, student, and researcher with better way to procure appropriate research tools and services. We also focus on the capacity building because it's very important to train the next generation of researchers and also share all the knowledge we've been able to develop. So we do that through various way, capacity building workshops, so that we really prepare young people to be able to use innovative solutions within their own context. And then we are involved in research. We were lucky to have funding from international organizations to work on concrete research projects. Sorry, we have one here in collaboration with various organizations, including Cameroon Ministry of Defense, on development of the lamb kit for COVID-19. Also, we are working on developing a new diagnostic for typhoid fever using the CRISPR-Dx technology. So now, it's very, very important for us to mention a bit about open source approach. It's a new kind of trend that is existing now, a kind of philosophy and a different way of doing science and research. When you embrace this open source concept, is providing some interesting solutions and advantages because usually open source approaches accelerate research and development and reduce the friction in access to knowledge and material because it promotes a lot information sharing and resource sharing. It means that at the end of the day, you have a lot of help when you want to do a particular innovative or very advanced experiment. User innovation, since it also encourages collaboration and multidisciplinarity, at the end of the day, the the products that come up is a product that was built with the consensus of many people and usually is a plus for the adoption. And also focus on value add because it's very important to I mentioned it's very important to target concrete solution, and open source approach are very good in that sense. So now how everything started for us through collaboration. I mentioned that collaboration is a very, very important aspect of this journey. At early stage, we started working with the Open Bioeconomy Lab, which is an international organization based at the University of Cambridge in the UK. We work on what they call the open enzyme collection. Basically, we work on the resources that were generated by the Open Bioeconomy Lab. So, which is basically a toolkit made providing a comprehensive list of information around enzymes. So if people are interested to produce a specific enzyme for various applications in molecular biology, so there's what they call the open enzyme collection that you can access through this website and through the Open Bioeconomy Lab platform. So it's really a comprehensive list of information you need to produce the enzyme. So it goes from the sequences of the enzyme that are going to be used in the vector to other information. So the innovation here was really to identify patent-free enzymes. So it means that after the protection, the copyright has expired, now the information comes to the public domain. So the team took the time to really identify the enzymes that were patent free So that it means that everyone is able to reproduce and produce it locally and now compile it into the open enzyme collection. It's a very, very important tool for people interested in local enzyme manufacturing. So we with the collaboration, we pioneer local enzyme production. So at one point, we became autonomous and able to produce locally a various set of enzyme. I'm going to share a bit about the methods. So now it's a bit technical here, but I will try to really try to explain it in a very, very simple term. Usually the process we follow to produce the enzyme locally. First, we get the construct of plasmid. So the plasmid is the vector or the tool we use to insert the particular gene of interest we want to express or produce. Usually we get it through collaboration again from partners. Now we do locally what they call cell transformation, basically inserting the plasmid into a bacteria. In our case, it's E. coli bacteria. And then we grow the bacteria, and then we go to protein induction using a particular chemical. At the end of the day, we have the protein expressed, protein which are in our case enzymes expressed. So the next step is to purify it, because at the end of the day, you have the protein and other cell debris. The bacteria themselves are in the culture media. So we need to isolate specifically the enzymes, and we do that through different protocols or methods. We have very pure enzyme through affinity chromatography, is stuck here. We also investigated very, very simple method for purification of the enzyme, such as ammonium sulfate precipitation. We also explored the cellular reagent technology that was developed by some collaborator in the US, basically using the bacteria without purification. It's very, very good, because at the end of the day, you have two different features and specific enzyme. We use that now to do a kind of R&D process to come up with new products. For example, after producing the enzyme, we can do master mix. So we just add other component needed for the PCR reaction, polymerase chain reaction, and then we produce our own master mixes. We can also produce new products after the R&D process just by identifying formulation and simplifying maybe the use format of particular products. For example, the buffers that we use for electrophoresis, you can just prepare it in a more user-friendly way, and it becomes a new product. So there are various ways of doing innovation at this point, but what we did was really, we did not reinvent the wheel, but we really identified and leveraged on the local production platform to come up with other products that will be useful for researchers and students. And at the end, we have new products. So now, within the course of doing that, we started developing our own protocols using low cost and available products and resources. Because one of the main challenge we faced was basically access to some fancy and advanced equipment. And we tackled that by really trying to use available resources. So we came up in collaboration with our partners as well, with protocol that enables, for example, the production of proteins on plates, on agar plates. Usually, protein expression is often done on liquid platform, but with this protocol, you are actually able to produce the protein on plate and overnight. So it's again in time, again in resources, and then the results are very as good as the liquid and conventional protocol. So we also demonstrated that it's actually possible to replace one of the most expensive chemical used in molecular biology for protein expression, the IPTG, by lactose. Lactose that you can get from various sources, including commercial, like commercially available milk. You can have it boiled fresh. As long as you have lactose, it's possible to use it for protein production in replacement of the IPTG. And then we also prototype some equipment that are needed for protein production. So as we are going to see on the next slide, all these resources are publicly and openly available to anyone who wants to replicate. So it's a very important aspect of our work to be able to share any innovation, any result we have with everyone. So now, after coming up with these R&D, so if you remember, we started with concrete innovation. We started developing our own protocol, like establishing the platform. But how did we do to translate that into a concrete company, a concrete product? So this is something very important. How do you move from the wet lab, from the research to actually coming up with a product that is commercialized through a company? So we've done that through collaboration once again with the UK-based not-for-profit company Beneficial Bio. So we became a biomanufacturing node, meaning that we produce locally the reagents that are commercialized in Cameroon, and we also had some sales in Ghana and Nigeria. But once again, the problem that we are trying to solve, and which is very important to keep in mind, whenever you want to go through biotech entrepreneurship, you really need to keep in mind the problem. In our case, the problem was recurrent context-specific issues faced by researchers. Like passing through this issue myself as early career researchers, it was very difficult to have access to some of the reagents and equipment that we needed to carry out very basic molecular biology experiment. So at the end of the day, we wasted a lot of time. Like some of the reagents were so expensive that it wasn't easy for us to procure. And then we had a lack of specialized services such as sequencing. It's just not available in the country. And then even when you have the resources to buy the reagent abroad, at times you have to wait for more than a month for the reagent to arrive in your country, and at times there are issues with custom and also cold chain maintenance. So at times the reagent arrives to you very bad in bad shape. So all this, like the result is people dropping out from school, like delays in carrying out research. You can spend years trying to conduct your research, and also it limit the contribution of African researchers, specifically as far as molecular biology is involved. So now we really trying to address this problem. We set up a more formal company, still doing what we started from the wet lab. So now, what do we do concretely? We produce and sell reagents and equipment to help innovator, public and private sector organization. So we are able to do the local production of some of the reagent, and then we also help like researchers, students, and organization to procure some equipment and other reagent that we get from trusted suppliers. So we do two things. The reagent that we are not able to produce, we get it from trusted network or suppliers, and that's how we are able to help universities, hospital, research institutions with innovative products and what they need specifically, the tools they need to conduct their research. So this is just to show the local products we are currently able to produce. Mostly, we focus on electrophoresis kits, like all the buffers you need to carry out electrophoresis, PCR master mixes, so which is a premix containing everything you need to do the PCR reaction. You just add your template and also your primers. We are also able to produce various sorts of enzymes, various kind of enzymes from the open enzyme collections I mentioned. So what is very important here is to set up the platform for enzyme production. In our case, it was a cell-based system, so we use bacteria to produce the protein. But there are many other platforms. But once you do that, the only thing you are going to change is the plasmid. And then some minor aspect of the protocol. But once you are able to set up the platform, it enables you with the potential to produce a lot of different proteins. So these are the products we currently make available to researchers locally. The focus on PCR products comes from the local context. So we ran an enzyme survey at early stage, and this is what the local researchers really were focusing on. They said they were very interested into basic molecular biology tools. So that's why we really started our production with these reagents. But now we are diversifying our production to many different enzymes applied for the industry, for example, the agro industry. Now we also have a lot of enzyme in environmental issues like biodegradation of some plastics and all that. So the potential is really, really huge once you have the platform set up. So now we also came up with some innovation in terms of the access and user relationship. We enable mobile money payment because it's very difficult for students, especially to have access to the bank account. So most often we interact with them and we enable payment of the products through mobile money. Also, we interact through WhatsApp and other innovative social media. So this improves the kind of customer support we can provide as well. So we stay available for any question, and in a sense, it's an innovation as compared to what's going on, what was going on before. So the local production here comes with a lot of advantages because the reagent are produced locally. It means that no shipping and custom issues. The cost is reduced because you master the production line, so you know exactly what to use to reduce the cost, and also um, it has reduced the delivery time. Now we are able to deliver products in less than 3 days within the country instead of waiting for months. So progress. So we now work with the largest health and research institution in the country. For some of the products, we have 50%, more than 50% in the reduction of cost of the good achieved through local manufacturing. And then for the time and the price, some products can have close to 90% reduction in cost. And then the time is 2 weeks for primers, for example, instead of months. So it's important to mention here that it's a step-by-step journey or enterprise. So, and for that, you also need to think about the resource and availability of funds. So in our case, we really identified what we needed and started at a very basic setting. As we can see, this is how the lab looked like in 2019. We just had what we needed to do to carry out very basic experiment like the local production and using open source, open science concept. In 2021, the lab evolved a bit. We now acquired through collaboration and partnership and some grants, more equipment to be able to do more experiment, more advanced experiment. And then we set up another lab only dedicated to the production of the enzyme, because when you follow quality standard, you need to separate anything that has to do with production with training. So we now have two different labs, one dedicated for the production and another one for the training. So we also set up in 2022 a bio innovation hub, basically a lab that is dedicated to training. Sorry, and as you can see, it's just a small video tour of the lab, how it looks like. So this lab is mostly dedicated to training, and we give access to students and researchers in need of carrying out their research projects. It was inaugurated in 2022. And then we have the production lab, the lab that is completely dedicated to the production. We can also have a tour, how the lab looks like. So it's a kind of step-by-step way. So once you start also redefining your strategy and adjust accordingly. Now we are able to work on innovative solutions. So this is, for example, the shaker incubator that we prototype locally, and all the information you need to replicate this particular equipment is available on GitHub and GitLab. So basically, these are some of the experiments we carry out in the lab. Okay, I mentioned the importance of having open educational resources because it's so important for us to share whatever knowledge and information we generate. So that's why I'm so happy to be here with you to share our experience and also some of the tools and resources we have and took the time to develop. So most often we share protocols or all the information you need concerning our products. We share them openly through this platform, the protocol.io platform. I'm sure some of you know this platform very well. It's a great resource, a great platform to share protocols and also collaborate on scientific resources. So most of the products that we commercialize or we make available to researchers are also what you need. The protocols themselves are available here on the beneficial bio platform on protocol.io. So I think I encourage everyone interested to have a look and also have access to these resources. For example, this is how it looks like. You can have all the information you need to produce the DNA polymerase. Also, this is the gel staining reagent for electrophoresis. So I really encourage people to have a look and provide feedback as well, and if the protocol works for them. We also share a lot of resources and protocol on BenchLink, which is a cloud-based platform for biotech and R&D. It's a very powerful resource that we use a lot to. So in addition to the access of reagents and enzymes, so it's also very important to be able to have some locally made equipment. So in our case, we prototype two equipment, a microbiology incubator and also a shaker incubator. So these equipment are very useful for enzyme production. And again, all the information you need to replicate and reproduce this equipment are found. You can follow the link to have all the information you need to replicate it for your own lab. So we also work on artificial intelligence. So now sharing the experience, I mentioned that training and capacity building takes a very important part of our work. So we really take the time to share all the resources, anything we develop. So it started by a technology transfer from the Open Bioeconomy Lab and many other organizations such as Beneficial Bio who helped us to set up the first lab. It was a kind of UK to Cameroon collaboration. So now we are able to do more trainings in Cameroon and other countries because of the set of the display and the platform we've been able to do. So now we provide training. We also give access to reagent and consumable to students. So mostly in the Bio Innovation Hub I mentioned, we welcome masters and PhD students for training and also to carry out their research projects. Also, we work with local universities to provide trainings on site. So we go to the university and also give some trainings on. So if like people interested in replicating this open hardware, so in addition to the equipment, we also investigate producing our own equipment in addition to the reagents. So I also encourage people to have a look to replicate in their own labs. Now, I mentioned that we are also interested to share whatever innovation, also the protocols we develop through different means. So after benefiting from trainings from collaborators in the UK, we are now able to share all the knowledge we generate with people in Cameroon and abroad. So we do that through our Bio Innovation Hub, which is a dedicated space to welcome mostly masters and postgraduate, masters and PhD students to carry out their research projects. So we give them technical assistance. Also, since we are able to produce locally, some of the reagents is more affordable for them. So this is one way. The other way is that we work with local universities. We have partnership and collaboration, enabling us to run workshops and also other capacity building activities using our own locally made products. It's very important for us because we can also get some feedbacks on the quality, and at the end of the day, it helps us improve the quality of our products. We also organize some specific capacity building workshop for targeted groups. So I don't know the case of Malawi, but in Cameroon, for example, we tend to encourage a lot women in science. So at times we organize workshops specifically dedicated to women, and then we go through theoretical knowledge and also hands-on practical workshops. So far, more than 150 students and researchers were trained through this process, and then from various local universities. We also organize early career inspiration workshops with high school students to kind of open their minds in the future and the potential of biotechnology. So it's very important because at early stage, people tend to be very innovative and also provide very interesting feedbacks. And at the same time, they get inspiration from our work and give them the opportunity to think about their future. Um, so yes, this is training ongoing at the Bioinnovation Hub. So now I mentioned that we are also trying to replicate the experience, share the experience through collaboration. So we work with other organizations to share whatever resources and protocols and hands-on practicals we have. So in collaboration, for example, this was in South Africa in collaboration with Trend in Africa organization. There was a workshop on open reagent manufacturing. Now we also have people interested in Nigeria. We also work with collaborators. This was at Covenant University, Nigeria, to assist researchers in other places, still on open enzyme manufacturing. Also, we had the opportunity to go to Southeast Asia, where we also train and help people setting up platform for local enzyme manufacturing in Indonesia. So there's a lot of attraction and potential for this particular technology, and we are happy to see that as time goes, people are more and more interested in being able to producing their own reagents, also to develop their own resources, because it gives a lot of autonomy and freedom once you are able to produce your own reagents. Now you can use it for either entrepreneurship if you want to commercialize or just for their training. Because if we are able to give this training, it's because we are actually able to produce locally. So we reduce the cost of training. So there's a huge potential. So now I would like to talk a bit about the challenges faced when you want to go through this process. So we have funding and grants. So it's always important to look for opportunities to be able to have access to the reagent equipment more specifically. So there are open calls for grant application. You just need to really work in collaboration to and also have innovative projects. So we are going to touch base on that a bit further. Also capacity building. So it's always important to look for opportunity to upgrade the skills and expertise for the team. So it's good to look for scholarship, conferences, research visit, and placement. Access to research facilities. So at times, collaboration is very useful when you want to develop particular innovative projects. So if you don't have access to the resources, you can work with another organization, for example. So now, one of the main challenge we face is first having more local support from our local organization, because so far in our own experience, it has been very difficult to have access to funds from the local government or regional organization, African organizations. Most of the funding we got were coming from international collaboration. So we always try to draw the attention of local organization that there's a huge potential in molecular biology, and then it's very interesting if they could support in any ways. Another very big issue we face and we've identified after developing all these products and resources is that at the end of the day, you also need to build trust to locally made products. I don't know if it's similar to your own setting, but in Cameroon, for example, um, people tend to rely or trust more the products that are coming from the West or from other European or American countries. So once we talk about locally made, in people's mind is that the quality is generally poor, but it's not the case. So at one point, you also need to also talk again about building the trust to your products so that locally the researchers and students are able to use them. That's why I mentioned that it's very important to involve them right from the beginning, because at the end of the day, you don't have any guarantee that your products will be adopted by the scientific and research community. So it's very important to involve them in the process, also organize a lot of activities where they can see what you are doing. So in a sense, there's a lot of initiatives all over the African continent about being able to produce locally different tools, different resources. But at the same time, we need to ask ourselves the question: are we actually ready to accept these locally made solutions? It's an open question, and in our case, we are still raising awareness about the quality of our products and everything. So, because at one point we need to really think about local consumption as well. So, to talk about the lesson learned, a more concrete way of what you need concretely to go towards biotech entrepreneurship. Like, I think you need these four elements before you start any biotech organization. The first thing you need to have in mind is the ideas, the innovation itself. Once you have that, you need a team. So the team has to be multidisciplinary. You can also have multicultural. So in our case, it was very, very useful to have people from different regions of the country sharing ideas. Passionate, because at the beginning is really difficult. So you need to really work out of passion, because the benefit and the profit tend to comes after some time of operation. So you really need to be dedicated and passionate. Also, the management style. Now, new management techniques involve like really empowering the team with leadership. So it's not like the formal way of hierarchized way of running organization, but most often for biotech ventures, there's kind of linear model where everyone is a bit at the same level, and in that way, it's much more easy to come up with innovations. So now, after you need to also think about resources and funds. So how to get physical assets like the equipment, even the local where you are going to build your company, anything physical you need. You have to draw a fundraising strategy according to your own sensitivities, because there are different ways of getting funds. So now, depending on your mission, vision, and values, you target some specific like funding opportunities. Also, it's good to have what they call the sustainability mindset. So, in whatever you do, you always need to think about the kind of operational cost of it. Always put in mind that at the end of the day, you need to sustain the activity, and for that, you need to be very pragmatic on thinking about certain operation. Like always think about how to sustain that. Not always focusing on the science part, but also think about the business side. You also need a community, so the innovation and creativity, like the community will give you feedback, which is very important to improve the quality of your products. Also for sustainability, because at the end of the day, the community is the users. We provide not only feedback, but they will be the one buying your products or using your innovation. So it's very important to make them and work with them, have them aware of anything you do. So communication is also very, very important. Network and collaboration, because collaboration will help you solve technical problems. Usually, it will speed up the processes of coming up with the innovation or improving any scientific experiment. It give you access to new opportunities because using through collaboration you can have access to training, you can have access to resources, to facilities, to opportunities in general. So and also it enhance credibility. I mentioned that the mindset, like for the case of Cameroon, is to trust more organization or products that are coming from like developed countries. So now, once you work with strong partners, it tends to help in the credibility of your innovation. So these are just some lesson learned from us. So now, um, to talk about like fundraising strategies, how you go through this whole process in a nutshell. The first thing is to develop a compelling business plan, because one of the major issues or pitfall at the beginning of biotech entrepreneurship is to focus mostly on the science part. At the end of the day, what you want to run is a business, so you also need to follow all the steps needed to run a business. So having a business plan, clear business plan with all the aspect of it is very important. I mentioned that you need to build a strong team. Also think about intellectual property protection, because remember that we are dealing here with innovation, with an innovative, a new products. So it means that you might want to protect your discovery, and there are mechanisms for that, trademark, copyright, so you can feel free to go. There are also regional organizations taking care of protection of innovations you can approach in addition to local organizations. You also need to conduct thorough market research. So at the end of the day, you need to know exactly who could be your potential customers and what they need, what they want, and also is useful because they will also provide useful information about the potential lacks of the current system that you can fit in and try to address. Uh, yes, at times you need to seek governmental grants or research funding. I mentioned that in our case, we receive more funding and support from international partners, but there are some local grants available. So we need to talk to the right people within your country and also draw the attention of the political leaders on the potential and benefits of biotechnology, because at one point it's our duty to educate and inform our policymakers, so that if there's a lack of funding or a breach in support, it's because maybe they are not aware of the potential of. So in part of our work is really to work hands to hand with political leaders and decision takers to really make sure they understand the potential, and also to see how they can support. So we involve them in trainings, we involve them in involve them in all the activities we run. We really want to have them around. So yes, I mentioned that it's very important to participate in research competition and also grant application to secure non-dilutive funding. As a biotech startup, there are a lot of competition where you learn a lot because you have people reviewing your ideas, curating it. So at the end of the day, you really come out with a lot of help. We can also have most of them at the end. You have access to some funding, or you have the opportunity to pitch in front of investors. So usually they are very good exercise. You can also engage directly with people who want to fund or startup. You create a targeted list of potential investors, and depending again on your sensitivities and what you want to do, approach one of the different kind of investors that exist. Attend in the industry conferences and networking. I mentioned that collaboration is very important. So usually you have the links, also the contacts of other company and also potential collaborators through conferences. So it's very important to attend these international events. Also, you need to build its strategic partnerships. So once you have the link, also with trust, you can really make strong connections, and that can help you through different aspects, technical, funding, and also other important aspects, regulatory aspect as well. So yes, you can also consider crowdfunding, where you have like the international community contributing to the success, or for you to have some funding to do a particular like carry out a particular project. There are many platforms of crowdfunding that exist. So it's also an interesting way of raising funds. You can also leverage on incubators and accelerator. This is mostly for early stage company. So you often have programs that help you build your startup from scratch. It's always good to record and show progress and milestone, because at one point, if you focus too much on the science part, it will be very difficult for you to know the key impact indicators that you need to really document, and to see the progress you make. So it's very, very important to keep in mind what are the various steps you are crossing, and also what particular indicator you might want to watch out for, so that you are able to trace the success, the evolution of your company over time. And then be transparent and responsive, because collaborations mostly involve trust, and it's very, very important to maintain that. So you need to be very honest, transparent. Like when you can do something, say you can do; when you can't, also be very clear with it. And also, yes, it's very important for fundraising as well. When you have a grant, it's very good to deliver on time, and be careful about certain things. And at the end of the day, be patient, because it's a process that can take time. But with the right support and help, you can definitely see through, or have a way out. Like any other organization, any other company, there are challenges, and why or not. So it's the same case for a biotech company. Yes, in a nutshell, what to really watch out for, you need to think about the end user first. So whatever innovation you want to work on, you want to develop, you need to think about who will be the one using your products, and also involve them right from the beginning. So you can do that through customer feedbacks, getting feedback from people, market analysis before even you start developing. So this is very important to increase and ensure adoption at the end of the day. Collaboration is key. So collaboration speed up processes, like enable to overcome technical challenges and provide opportunity for growth and diversification. So it's very important to find collaborators. So there are extensive list of organization, and I mentioned attending events actually enable us to have access to great collaborators, and you can design very interesting projects. So everything is possible with a great team. So like organization are made by people. So it's very good to find the right people and also know how to motivate and also keep them, so that your company will grow, because usually it's one of the main reason for failure, when you are not able to identify the right team, and you don't have the right strategy to keep them. So you need to think about your career evolution, and also yes, also have the mechanism to attract new talents. So I think it's also critical to think about. Yes, so I think I will stop here for now.